Most drug fails not because they don't work, but because of ADME. I just read a paper that shows how simply replacing a phenyl ring can transform a molecule's entire drug profile. Phenyl rings are flat, aromatic, and because of that, they can oxidize and form toxic metabolites. This paper replaces them with 3D saturated bicyclic structures, same geometry, but no toxic bioactivation, better stability, and improved solubility. Results up to 58% better metabolic stability and 100% oral bioavailability. This is not just chemistry. This is how a molecule stop being just an idea and start behaving a medicine. Either go to the paper, read the paper, or do continue with the video. All the very best.
Students, today's paper is again from medicinal chemistry. One second, let me select few things so that I can write here. Okay, so medicinal chemistry paper. Very interesting paper because here they have chosen this molecule, saying that here they are going to do what? They are changing this aromatic part to these biocyclic parts, and they have found a very you can say great and considerable changes, not changes, but improvements of the same moiety for biological activation. Okay. I mean, they have shown better biological activities, okay, compared to the molecule when it is containing phenyl ring in place of these bicyclic rings. Okay. Now, why I found this paper very interesting, because they are going from flat to 3D model. Okay. Now here, let's start from the scratch. Let's start from the this title of the paper: replacing phenyl rings with saturated bridged bicyclic mimetics to mitigate bioactivation. Bioactivation is mitigated and optimize ADME properties and over bioavailability in reverse amide orientation. Okay, so here this is reverse amide. Okay, so first of all, this amide presence and amide orientation is very important here. Okay, first of all that. Secondly, second point is they're going from flat to 3D. Okay, flat system to 3D system. Now 3D, first of all, what was the problem with aromatic rings? Okay, what was the problem with the aromatic rings? The problem with the aromatic rings was aromatic rings are flat. Okay, flat. They are electron rich. Okay. That's advantage as well as disadvantage. Okay. Now three flat problem is if the target is flat, in that case this is good. But if the target pocket is 3D, then if your you know your molecule, your drug is also 3D, that's going to be a plus point for you, that it can interact with different amino acids present over there, and your drug can reach in a better way. Okay, and the target can be targeted in a better way. Okay. Okay. Okay, so this is the this is the importance of their 3D model. Okay, they're electron rich meaning they are highly lipophilic. Okay. Okay, sorry, let me write it down properly. You can increase the speed of the video and you can jump to the further part of the video. Okay, lipophilic. Okay, when But the problem with lipophilicity is when your drug is highly lipophilic, it will attract to proteins. Okay, proteins like membrane. Okay, so it will kind of stuck to protein. Okay, so I uploaded this short in which I have told you the the importance of logarithm p of drug. Okay, so I have shown you the formula, and there I have told you that this logarithm p value should not be lesser than 1 and greater than 3. Around 1 to 3 is the ideal value. This is enough permeable and enough hydrophilic. So if you have a drug with high lipophilicity, it will stick to the membrane only. Okay, it will not enter to the cell. If you want it to enter to the cell, it will not able to enter the cell. And if your drug will not enter the cell, if cell is your target is inside your cell most of the time, okay, then it's going to be a big deal for you. Okay, so it won't be easy for you to penetrate the membrane if it is highly lipophilic. Okay, next is they are metabolically vulnerable. Okay, metabolically vulnerable. Okay, they are metabolically vulnerable. Metabolically vulnerable meaning if it's present there. Okay, there are different kind of oxidation process that it can undergo. Okay, it can undergo epoxidation. Okay, it can undergo quinone formation. Okay, quinone formation, it can undergo oxidation meaning quinone formation, and different kind of other possible oxidized product. Okay, so that's why it's very these points makes the presence of phenyl ring not like that likable for the drug. Okay, now let's jump into the abstract now. Replacing phenyl rings with saturated bridged bicyclic scaffold can mitigate liabilities inherent in planar aromatic rings. So they are saying if you replace phenyl ring by bicyclic scaffolds, the inherent liabilities, these are the inherent liabilities, they can be avoided. Okay, in the first line they are saying that these liabilities can be avoided. Okay, now next line they are saying we evaluated the electroisosteric impact of retroisosteric impact. Okay, retroisosteric impact meaning replacing the kind of a big part of the drug with something else. Okay, that's retroisosteric isosteric change. Okay, the impact of phenyl to bridged bicyclic substitution on bioactivation and ADME PKA using phenyl prototype 2B that displays rapid microsomal turnover, glutathione adduct formation, high plasma protein binding, poor solubility, da 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 da. Okay, now what are they saying? They're saying that what we did, we thought of a molecule. Okay, we found a molecule, not thought, they found a molecule. This they named it as 2B. Okay, now these are the problems that were faced by 2B. Poor binding, high plasma protein binding. Why? Metabolic vulnerability, lipophilicity. Lipophilicity. Okay, it's going out of the screen. Okay, lipophilicity is high, that means protein binding will be there. Okay, poor solubility, they are poor soluble in your water, aqueous medium of your body. Okay, so it is very high to kind of soluble them in blood, and so that it can reach the target. Okay, they are they display microsomal turnover, meaning they kind of you can say metabolize CPY 450 enzyme. Now, for okay, this is the enzyme, and they can kind of uh, metabolize. Okay, okay. What is glutathione? GSH they call it. Okay, glutathione GSH, and they can form adduct with glutathione. Okay, glutathione adduct formation. They can form adduct with glutathione. That is glutathione adduct. Okay, so this causes toxicity. Okay, meaning they chose a molecule with different kind of problems which were already known. Okay, they didn't choose any drug molecule that has no problem or has minimal problems. No, they chose a molecule which has many problems and solved it. That's the gist of that's the heart of the paper. Okay, heart of the paper. Okay, now let's move on. Now here they say bridged bicyclic analogs abolished. Abolished meaning terminated. Okay, completely removed. Reactive metabolite formation. No metabolite formation is there. Okay, improved metabolic stability. While bridge had substitution delivered greater improvements. They're saying that simple bridge bicyclic substitution kind of gave it gave really improved results, but when you substitute them with like fluorine, oxygen, okay, that improved the that showed that displayed improvements 58 folds, 58 times improvement was there. Okay, only 2 to 6 fold is great result, but they are showing how much 58 fold, 58 fold improvement in mice. Fluoro BCHEX improved bioavailability to 30%, whereas oxa BCHEX achieved 100% bioavailability. What is BCHEX? Bicyclohexane. Bicyclohexane. This is BCHEX. Okay. Now, bicyclohexanes, what they did, they improved the bioavailability 10 times higher exposure than 2B. Okay, they shown higher bioavailability. Okay, now this is great. Okay, that means if you take bicyclohexane and substitute it with fluorine or oxygen, this is going to give you great results. Okay, next is MMP analysis. One sec. Now here in this line, they are showing, they are kind of trying to tell us that experimental part, okay, little bit experimental part, that MMP analysis. MMP analysis is matched molecular pair, molecular pair, okay, matched molecular pair analysis showed that reversal of amide bond, this amide bond plays great role. If you reverse it, CO here and NH here, then the results were not really good. In my mouse liver, MLM half life lowered. Okay, MLM half life lowered. MLM is mouse liver, mouse liver metabolism. Okay, so it got lowered. If it's lower, that means this this will be worse stability. Okay, this and this drug will be, so sorry, here the metabolism of this drug will be lowered. Okay, which is not the case of human liver, however. Okay, human liver permeability was unaffected. So like it depends, what kind of study are you making. Are you making the study in cell assay? Are you making the study in a mouse? Or you are making the study in humans? Okay. These findings position bridgehead of bicycle as effective benzene mimetics for mitigating bioactivation with incremental of EDME effects. EDME is absorption is better. Okay, absorption, distribution, distribution is good. Okay, metabolism, metabolism is also nice. They do not get they do not metabolize microsomal turnovers. Okay, now excretion, they do not cause microsomal turnovers. Okay, and excretion was also good. Excretion, please pardon my writing. Actually, I'm very new at writing on a screen. So yeah, pardon me for that. I hope you are getting what I'm writing here. Okay. Okay. So all these things were really good over here. And this paper I really liked because it gives you really a good insight, or you can say not insight, but it gives you really good idea. If you're working in this kind of this kind of if you're working with this kind of rings, you can replace them with bicyclic these bridgehead functionalizations, and you can lead to great results, better results. Okay, so yeah, so I really like the paper for because of that. Okay, so also the synthetic part is very uh, synthetic part synthetic part is also very easy. You can take these bicyclic rings. You can take these bicyclic rings acids here, COOH. However, they are very hard to synthesize in lab, but you can find them commercially available. They will be very definitely high priced. Okay, but your results will be great. Okay, so you can go for that. So I hope you also like the paper, and you can use the paper, the idea for your research. And if you want to read more about it, go to the go to the paper, go to general medicinal chemistry, and read the paper. Also, these videos that I make, they are not sponsored by Journal of Medicinal Chemistry. I just like their paper most of the time, and yeah. Okay, all the very best. Thank you.